Students, we finished our last regular exam, and what we do from now on will go toward our final exam. About 30% of the final exam will actually be the material that we cover from now to the end of the semester, and the rest of it will be on what we've had in the past. In order to follow along in Chapter 13, which is the spinal cord and spinal nerves, I'm asking you to make sure that you have printed your spinal cord, spinal nerves document and have filled that out, or at least started filling that out, because I'm going to go over that as we go through Chapter 13. All the time we hear of individuals who've had spinal cord injuries, and those can be devastating. The spinal cord, which is a part of the central nervous system, is actually the connection to the brain from the body. And in this chapter, we're going to look at spinal cord and spinal nerves. On the first page of your document, I've asked you to list some principal functions of the spinal cord. Those include conduction. That means we're going to have fibers that are conducting sensory and motor information up and down the spinal cord. We have neural integration. That is, spinal neurons receive input from lots of different sources. Then they integrate it, and then they come up with an output. Example given, bladder control. Locomotion. Your spinal cord contains central pattern generators that you're really and truly not aware of, but those are groups of neurons that coordinate repetitive sequences of muscle contractions so that you can walk without falling down. And then we have reflexes. These are involuntary responses to stimuli, but they are vital for your posture, for your coordination, and for protection. I did not include sensory receptors on your document, but I would like for you to be aware of these. Sensory receptors. These are specialized to respond to changes in the environment. Those changes are called stimuli. And the activation of the receptor ends up with a graded potential that's going to trigger an action potential that's going to be a nerve impulse to carry that information. There is a difference between sensation and perception. Sensation is going to be the awareness of the stimulus. Perception is your interpretation of that stimulus, and that's occurring in your brain. I want to talk about how we classify those receptors. They're based on the type of stimuli they detect, their location in the body, and their structural complexity. We are going to focus on the stimuli they detect and the location in the body. Here's the classification by the type of stimulus. And the names pretty much tell you what they are. Mechanoreceptors are receptors that respond to the mechanics of touch, pressure, vibration, stretch, etc. Thermoreceptors, they detect temperature changes. Photoreceptors respond to light energy. That's what your eye is using. Chemoreceptors respond to chemical changes. That's why you have smell, that's why you have taste, and they also pick up changes in blood chemistry. Then we have nociceptors. Nociceptors are sensitive to anything that causes pain. When we have classification by location, we have exteroceptors, and those are going to respond to stimuli coming from outside of your body. So we're looking at the skin, the touch, the pressure, the pain, the temp, and your special sense organs, because those stimuli come from outside the body, like your hearing, like your sense of smell, etc. The other type, interoceptors, can also be called visceroceptors because they are responding to stimuli that is coming from your viscera, your internal viscera, and your blood vessels. They're going to be extremely sensitive to chemical changes, tissue stretch, temperature changes, and these sometimes are hard to actually pinpoint. You will feel a discomfort, but you honestly cannot pinpoint exactly where that's coming from. Then we have, so very important, the proprioceptors. Proprioceptors respond to the stretch in your skeletal muscles, your tendons, your joints, ligaments, connective tissue coverings on both bones and muscles. They inform the brain of your position and your movements, and that's why you can stand straight without falling over. Your survival depends upon both sensation and then the perception of that sensation. So the sensation, again, is the awareness of the changes in the environment. The perception is understanding what that is and interpreting that. To produce a sensation, we have receptors. Those receptors are specific for particular stimuli. The stimulus then must be applied within that receptive field. Think about me shining a flashlight in your ear. That does nothing as far as your hearing or your seeing. After the stimulus has been applied to the receptive field, we have what's called transduction, and that stimulus is going to be changed to a graded or local potential, and then that graded or local potential must reach threshold and create an action potential. I have asked you to record some characteristics of the spinal cord in your document on the first page. Please know that your spinal cord is well protected. It's protected by bone. It's protected by meninges. It's protected by cerebrospinal fluid. The spinal cord itself is simply a cylinder of nerve tissue. It's going to occupy the upper 2/3 of the vertebral canal. Most people think it goes all the way to the end of the vertebral canal. It does not. The very inferior margin is about L1 or maybe L2. The thickness of the spinal cord's almost 2 centimeters and about 45 centimeters long. This is in adults. It gives rise to 31 pairs of spinal nerves, and then we name the segments of the spinal cord according to those spinal nerves. We have as well longitudinal grooves. They're both anterior and posterior. Anterior and posterior we would also call ventral and dorsal. The anterior ventral median fissure is a groove. It's called a fissure because it's a relatively deep groove. The posterior or dorsal median sulcus is a groove, but not nearly as deep. Your spinal cord is going to be divided into, here are the sections, the cervical, thoracic, lumbar, and sacral. There are 2 areas where that spinal cord is enlarged. We have a cervical enlargement that's going to supply nerves to your upper limbs. We have a lumbar enlargement that is going to supply nerves to the pelvic region and the lower limbs. We have what we call a medullary cone, and that's simply where the spinal cord tapers to a point. And it's inferior to the lumbar enlargement. We'll look at a pic shortly. Then we have the cauda equina. That's a bundle of nerve roots that occupy the vertebral canal, and that's from L2 to S5. Here's your spinal cord, and here are the regions. This is the cervical region. Then you have the thoracic region in here. Here's your lumbar region, and then here's your sacral region. The two enlargements are shown here. This is your cervical enlargement, and this is your lumbar enlargement. What you see here is the conus medullaris. That's that pointed area at the very inferior of the spinal cord. And then you have here the cauda equina, named because it looks like a horse tail. And these are the nerve roots at the inferior end of the vertebral column. If we were to do a lumbar puncture or tap, that puncture needle goes into this space down here between L4 and L5. Remember, in this area, we do not have spinal cord, and so we do not take a chance of injuring the spinal cord. Here is another pic of the spinal cord. You see that we have that cervical enlargement here. We have the lumbar enlargement here. This is where we have cervical spinal nerves, thoracic, lumbar, sacral. You have your conus medullaris. You have your cauda equina. And then we have the filum terminale. And these are just fibrous extensions of the conus. They're covered with the pia mater that we're going to talk about. And they actually anchor the spinal cord so that your spinal cord stays in place. What you can't see here, but we can see it on a slide down the road, are denticulate ligaments, and they extend from the pia mater to the dura mater, and they secure everything in place there. I have asked you to list on your document the meninges of the spinal cord and the related spaces. What you need to know are meninges are fibrous membranes. They enclose the brain and the spinal cord. They're going to separate the soft tissue of the central nervous system, and the spinal cord is part of the central nervous system, from bones. From superficial going into deep, we find the dura mater, the arachnoid mater, and the pia mater. The dura mater, or dura sheath, is going to surround that spinal cord, and it is separated from the vertebrae by an epidural space. That epidural space is going to have fat and blood vessels in it. Then we have the arachnoid membrane, and that adheres to the dura, but it is separated from the pia mater by a subarachnoid space. That subarachnoid space will have cerebrospinal fluid in it. That's where we looked at that picture of the spinal tap, where that takes place. Then the pia mater is very delicate. It's a very delicate membrane. It is tightly held against the spinal cord, and it continues inferiorly, like I was showing you in the pic, as the terminal filum, or the filum terminale. It fuses with the dura, and it forms that coccygeal ligament. Okay, let's look at this pic. You can see the vertebrae. You can see this is a cervical vertebrae. We look at the meninges. We have the dura mater. That's very thick, and that's that outermost. Then we have the arachnoid mater, which is deep to the dura mater, and then we have the pia mater that is really tight on the actual spinal cord. Now, you see this area is the epidural space. It's between the vertebrae and the dura mater, and it's going to have fat and blood vessels in it. Then you see we have the subarachnoid space in here, and it's between the arachnoid mater and that pia mater that is very tight on the nerve cord. Here you can see the denticulate ligaments that hold things in place. We're going to talk about the ganglia, etc. later. These are the meninges. Here you see them again. We have dura mater, and you can see how that's a very thick sheath. Then you have arachnoid mater, and then you have deep to that the pia mater. You can see the pia mater in pink up here. And you can see this anterior or ventral median fissure and how deep that is. And here is the dorsal or posterior median sulcus. Here again is another view. Let's just look at this. Epidural space with the fat and blood vessels. We see the dura mater. We see a little subdural space in this pic, and that's deep to the dura mater. Then we see the arachnoid mater, and then we see the subarachnoid space, and then we see the pia mater, which is lining that nerve cord. I have asked you to describe the pattern of gray matter and white matter in the spinal cord, and here's a cross section. This darker area is the gray. The pattern in the spinal cord is there is a central canal. It is surrounded by gray matter, and then the gray matter is surrounded by white matter. In this central canal, you will find some cerebral spinal fluid, probably some ependymal cells with the cilia to move that cerebral spinal fluid. We have a connection between the two sides of gray matter called the gray commissure, and we have fibers that connect those. Now, what I want to show you is the pattern of nerve transmission. We're going to go here. Here we have an afferent neuron bringing in a sensory impulse. We have lots of afferent neurons bringing in sensory impulses. This is what we call the dorsal root ganglion. You'll recall a ganglion houses the cell bodies. So we have sensory neurosoma in here. And then the axons are going to travel through these rootlets and enter the nerve cord. And they're going to enter into this dorsal horn. This little section here is a dorsal horn. You have a dorsal horn on both sides. This is gray matter. Now that impulse comes in. That axon will communicate with interneurons. The interneurons will communicate with motor neurosomas that are in this large area here called the ventral horn. There's a ventral horn on both sides. And then that motor neuron or the motor neurons will send the impulses via their axons through these rootlets and back out to the effectors. So we have dorsal horns. This is where the sensory information is coming in. We have ventral horns. In the ventral horns, we're actually going to have the cell bodies of the efferent neurons. Now, you'll also note that we have these lateral horns. We have lateral horns only in an area of T2 through L1. And anywhere else in the nerve cord, you will not find those distinct lateral horns. Those lateral horns are specifically for the autonomic nervous system, for visceral information. So, what we have, again, sensory dorsal root ganglion, into the dorsal horn, interneurons, motor neurons in the ventral horn, information back out. Lateral horns are autonomic. Then we have white matter. This white matter that surrounds the gray matter. The white matter is traveling up and down the nerve cord. This white matter is myelinated, as you know, and that is bundles of axons that are called tracts. Remember, if it's in the central nervous system, it is called a tract. And we divide those into the dorsal areas. This is called the dorsal funiculus. This is called the lateral funiculus. And then the ventral areas are ventral funiculus. You can also see in this picture things you already know: your dura mater, your arachnoid mater, your pia mater, etc. This pic may make this a little more clear to you. What we have here are somatic sensory neurons coming into the dorsal horn. We have visceral sensory neurons coming into the dorsal horn. Here's where their cell bodies are, in that dorsal root ganglion. Then we have the interneurons in here. Then we have, as you can see, our ventral horn motor neurons. And these are the somatic motor in red. And then you have the visceral motor, which again are autonomic nervous system, and those are in yellow here. This pic is showing you the tracts of the funiculi. So we have ascending tracts, that means they are traveling toward the brain, and those are shown in blue. And then we have descending tracts. Those are traveling from the brain down the spinal cord, and those are shown in red. This is one of the pics that I asked you to label. This is out of your text. This is pretty much something that we've covered now up to this point, with the exception of pointing out what composes the actual spinal nerve. Remember, there are 31 pairs of spinal nerves, and what you see, or what we talked about, is the input and then the output. The input through the dorsal or posterior root, the output through the anterior or ventral root. And where those join, where those are together, right here, is the spinal nerve. Then that spinal nerve is going to divide again very shortly, and we'll get that down the road. But that spinal nerve is really quite short. This again is something that I asked you to fill in on your document. And as you can see, the site of efferent soma is going to be right here. These are those motor neurosoma, and so that is going to be C. Your sites of axons and afferent neurons will be at your dorsal horn, B. Your site of sensory soma, remember those are in the ganglia, so that is in the dorsal root ganglion, that is E. Your gray commissure is this connection, that's A. The horn that contains autonomic motor neurons is going to be the lateral horn, okay, that is D. And then the site that contains the central canal right here, and that is A. Spina bifida is one of those things that goes wrong. It's a congenital defect, and it's where one or more of the vertebrae don't form a complete vertebral arch to enclose the spinal cord. It's amazing that it's one baby out of 1,000. The most common is in the lumbosacral region. The most serious is spina bifida cystica. What we know now is that folic acid, which is a B vitamin, is vital for preventing spina bifida. and it should be in the diet of all women of childbearing age. Here's a pic of Spina bifida cystica. Coming in the next part will be spinal nerve anatomy, and then more as we continue through chapter 13.